So good afternoon, everyone. It's my pleasure to host this very first session of the CuttingEEGX event dedicated to methodological tools to provide insights either from EEG or from MEG data. So my name is Marie-Constance Corsi. I'm an honorary research scientist at Paris Benin Institute here in Paris. It's quite cloudy here, so it's like perfect proof that we're in Paris, in France. So as Max said before, the aim of these sessions is to provide an overview of the evolution of the research in this field, but also the next challenges and the opportunities for the next future. So again, here, each presentation will last about 15 minutes, followed by 5 minutes of questions, and then we'll have 30 minutes discussion session. Again, don't hesitate to post your questions on the chat. Of course, I can't promise that we are going to ask all your questions, but we're going to provide you at the end also an FAQ document with all your questions and also answers by our beloved speakers who will dedicate time just to answer them. Also, in the forum, you will find a post dedicated to this very special session on methodological aspects with the link to the dedicated GitHub page that gathers the slides presented today by our speakers and some little surprises, but I can't tell you. You have to go there. So, okay, now let's start with our first speaker, Benedikt Ehinger, who is a tenure track professor in computational cognitive sciences. Hi, Benedikt, in Stuttgart in Germany. So today he will provide us a guided tour of the regression models dedicated to EEG data.
Thanks very much. We will today discuss what I call the modern EEG regression toolkit. And of course, I can't cover everything, but I will cover 4 examples of this toolkit. In the end, it all focuses around this formula in the middle here. Perfect, but I promise you this is the only formula in the talk, and I have only 15 minutes, so it will be a little bit fast, but there's a recording available, and you can ask plenty of questions. We will discuss the general rERP or regression ERP framework. We will discuss overlap correction or deconvolution. We will discuss time continuous predictors, and we will also discuss mixed models. Right? Okay, let's get started. Right. So regression ERPs. So let's imagine you're doing a very standard experiment, e.g. eye tracking combined, because this is something that I really like to do. And you ask people to watch, to look around on the screen, and they make eye movements on faces and bikes. And you're interested in, let's say, whether bikes and faces are processed differently in the brain, right? And now you could just calculate an ERP of faces and ERP of bikes and compare these two. But there's a problem, right? If you allow this kind of framework, then you also will have different saccade amplitudes. So people will make small or large eye movements on and potentially biased. And also fixation position, maybe there are more bikes on the left side than on the right side, etc. So in a normal averaging procedure, you can't take this into account. But you can then, well, maybe you heard somewhere that taking a mean is just a very special type of linear regression. So we're going to do that. In the end, you will never actually need to write your own, what you will see in a second year design matrix, etc. But you will always use this simple formulas like you maybe know from R or something like that. Okay, so what do we have here? Here we have single trials, so face versus bike trials, right? We select one of these samples. They are time locked to the, let's say, onset of the fixation. And now we build a linear regression model here. And this linear regression model, this consists of a design matrix here and some parameters and some error. These parameters are the ones that we will save for each iteration. You will see in a second. And this design matrix is special because we can modify it in many different ways. So in this case here, it is modified that we're going to find a normal ERP here, and here we're going to find the difference of what bikes are compared to faces. There are many ways to build these. I don't have time to go into that, but I'm very happy to discuss. What we're going to do next is that we're going to repeat this for each of the time points. So we slide this over, and we save the parameter estimates here. And this gives our ERPs or our slopes, or in this case, difference waves, right? So that's easy. Now we can modify this design matrix. So keep this in mind here. And we can add more columns to it. For instance, we could add a column for saccade amplitude and a column for fixation position. And then we also get 2 more parameter estimates. We get 2 more parameters over time, as you can see here. And these are now not the classical ERP. This is a mean versus not a mean. But these are more like slopes. So I changed the saccade amplitude by 1 degree. How much does my ERP would change? Now, this is not enough for this actual use case, because what we're assuming here is that everything is linear. So there's a linear relation between saccade amplitude and the EEG. which maybe, as I pointed here, so let's put here saccade amplitude here. This would be the linear relation. Maybe this is the P1 or something like that. This would be a linear relation, but maybe the underlying relation is actually logarithmic or quadratic, or can be any kind of shape. So instead of fitting a single slope, what we can do is we can basically tile up this saccade amplitude predictor in different overlapping predictors, find the correct weights to add them up to this black curve here. And with that, what we're calling nonlinear splines or spline regression or GAMS, with this we can then model more or less arbitrary nonlinear relationships. And that's very nice. We can stack them together. If we look at the design matrix again, because I always feel this is helpful to see, before we had this one column here, and afterwards, we just have a set of different columns. So this is a set of different parameters. And of course, we also get a set of different parameters. How we continue on with this is a question for the next talk. So please invite me. And I'm happy to discuss that. To give you an example, this is work with Olaf Demigen. It's a free viewing task. So you just show natural images. And we look at fixation. So 0 is the fixation onset. And this is now split up by saccade amplitude. So we have here a nice saccade amplitude spline. We also have circular splines and all the fancy things, but I'm not going to discuss that. What I want to highlight here is that for the first 5 degrees of saccade amplitude, so very small to 5 degree, you have a large difference. But for the last ones plotted here, between 10 and 15, you actually have basically no effect. So this is a very clear logarithmic effect here. But if you just would move 100 milliseconds later, then you would see here there's actually a linear effect. And so it's quite interesting and I think extremely underexplored. Okay, this was the first feature kind of, and now I'm going to go for the next one, which I think is also, they're all, I like them all equally. And so this is, the problem is the problem of temporal overlap. So what do I mean with this? If we have a single channel EEG like this here, in our setup here, we have eye movements around 200 milliseconds. So that's very fast, but the actual ERP responses are longer than 200 milliseconds. So they will overlap in time, right? It's, of course, a simplified model, but it's very productive, you will see in a second, right? So what we're measuring is the overlap. And now we would like to come back to the unoverlapped ones, because here you can see the unoverlapped one, and now I add in the overlap, you see the shape completely changes, right? And this can be biased if you have different bias. And now you think, yeah, okay, but this problem is not relevant to me. I don't do EEG and eye tracking. I just do EEG, right? But then also this problem, you will see also in a second, is highly relevant. So for instance, you have stimulus onset and you have button presses. And sometimes you have faster trials, sometimes you have slower trials. So the overlap will also systematically vary. So you also, there are many cases where this becomes relevant. To give you an example, first I explain how it works. Now we're still in the linear regression framework, so this is cool, but there's an important change. So before every time point was fitted separately, but now we need to fit all the time points at the same time because they can influence each other, basically. So let's take the EEG now from top to bottom. This is now the continuous EEG instead of having epoch data. This is it's a big thing, right? We are not cutting data anymore. We are immediately modeling the continuous EEG data. We have the different event onsets here as the bars. This is our Y, so what we want to explain. So we explain the single sample. So now we get a very big linear model. And as I mentioned before, now we have to fit all the parameters at the same time, right? So we have the long parameter vector, of course, typically not 10 parameters, but more like 1,000 or so, let's say, by 500 hertz. And we have a very similar to before. We have intercept and continuous predictors. And for each of the events, we get this kind of staircase. This is kind of similar, like this reflects where you are in this curve, in this local curve, in this local time. And now there's no overlap, right? So this is a bit boring. So let's add some overlap. So we move the events. You can see here, we move the events closer together in time. And now you can see that one sample is explained not only by a single event, but by the previous event as well, right? Or maybe even in some cases, there could be multiple more events explaining a single EEG sample. If you invert this, because all the events are typically not completely fixed, but there's some jitter, then you can invert this and you can recover the beta parameters. In practice, can look at something like this. This is work from Anna Gerd. She's interested in phase perception in mobile and moving, or like less restrictive situations. And if you just do the normal ERP, basically, so we don't use this deconvolution. We have a nice toolbox. You check it out to do this kind of things easily. If you don't do this, you find all kinds of differences. But once you apply the magic of overlap correction, what you can see, the curves pretty much overlap completely, except at the faces, right? So at the 170. So this is exactly what we, hope to find in more naturalistic settings. Another very recent example just came out a month ago or so from Romeo Frömer. She's interested in decision making, and there's this CPP effect where you have basically like a measure of evidence integration. So you have a faster build up for easy decisions and a slower build up for slow decisions until a threshold, and then you make a button press. And it turns out that if you apply this deconvolution methods as well, then there's no real difference here remaining, at least nothing significant. There's ongoing discussion. So there's a commentary, check it out, it's on bioRxiv already, that challenges this method. And I think, yeah, it's still up to debate whether this is something that we're allowed to do. But nevertheless, I think the difference is striking. And this is at least something to think twice about. Okay, let's go to the third from 4. Add-ons. This is what I call temporal response functions. So we so far saw that we can cut the data and we can just take the averaging, like the mean or the regression ERP. We can do this event-based deconvolution where we explain the continuous EEG, but there's another class when you have a continuous predictor, right? That is basically as long as the EEG signal, also sampled similarly, for instance, could be the envelope or the speech envelope. Then we can also try to relate this to the EEG data, and we can try to find out with which lag, basically, do we need to move this to explain some kind of EEG data. There in the literature, like I typically distinguish between these onsets kind of what I call rERP and the TRFs. Other people don't, so yeah, take this a little bit with a salt of grain. This is not something that's defined. Okay, how do we do this in the design matrix? Let's say we take one part of your old design matrix, and now for the TRF, we basically just add the signal here to the design, and then we time shift it by one low, one slower, one slow, one slow, low, and with all these time lags that we have here, we can then find out how the brain reacts to this continuous stream of changing information. Right? Because these will be typically highly correlated because there's autocorrelation in the signal. We need some kind of regularization now. And yeah, I don't have time to discuss that. Please ask me. I do have a slide on it, a short one. There are some cool regularization methods people are using. How does it look like in practice? This is from the eelbrain toolbox, and this is a spectrogram. So they were listening to some audio book, and this is the envelope, so kind of like the loudness. And we now want to relate the loudness to the EEG. And what comes out here in a type of butterfly plot for each of the channel would be different weights over time, how they change in relation to this continuous ongoing stimulus. So here, this is nice because we're moving away from this onset-based thing, and we have more like continuous signals that come in, come out. Okay, finally, linear mixed models. This is the fourth example I want to give. And I will give you I want to give you an intuition of why we need linear mixed models in the first place, right? So let's imagine you measure data, P100 or something, from a set of subjects here. Each subject, you take the average and you get 5 P100 values. You're not interested in these 5 values per se, but you're interested in the population response. So how do all people respond to it? And you get an average of this, and you get an uncertainty of this, right? We can calculate the variability. You get an uncertainty of that. Then a colleague comes in and tells you, hey, why are you doing this like that? Why you have, for each subject, you have 10 trials, right? Why do you average them first? What I always do, I just use all of the trials, right? And you say, okay, cool, I'm going to try that. Oh, this is awesome. I get much smaller standard errors. So this is really nice. But you're not allowed to do that, right? I'm just making sure. Probably you all know that, but if you do this, your p-values are too small. And the interesting bit I want to say is why are they too small, right? And they are too small because if you know already, if you know 4 trials of one subject already, and you find a 5th trial, then you're not getting complete independent information, but you kind of know already it's going to be on this side. It's not going to be on that side, right? If all others are here. So each subject kind of has like its own sampling distribution, and our intermediate conclusion is that the data points from the same subject, they are more similar. So we have to model it. We have to include that in the model. That's why we do within subject statistics, etc. Works out quite nicely. Now I could color these in a slightly different way like this. And how is this coloring coming from? This is coming from because the subject all saw different stimuli. So maybe they saw a sloth, or maybe they saw a giraffe. And if I show pictures of giraffe, then generally the brain response or reaction time or whatever will be more similar for all the giraffe pictures than for the sloth pictures. Not always the case. There's not always item effects, but they are also, the same conclusion holds. The data points from the same items are more similar, so we should also model this. This is very typically taken into account, and we generalize over subject, but this is very rarely. If you're not a psycholinguist, then item effects are super rare to be used. So how do we do that practically in EEG? Well, we have to change a few things. We have to first concatenate all the data of all subjects. Then instead of a regression, we have to use a linear mixed model instead of a linear model. And then in the end, we get not only fixed effects, which are like the group averages, the ERPs, but also random effects, which give us the variability. And just a very short preview of how this could look. This is implemented in Unfold's stats and in Unfold. Here, this is from the ERP core N400 task. It's just 2 words, and they're either related or unrelated. And we're looking at the second word and split it up whether, well, they are related or unrelated. And this gives a nice N400. And here I added subject effects. So this is the variability of a subject. As you can see in the beginning, there's pretty much no variability because it's baseline. There shouldn't be any difference. And then we get variability creeping up. But we also see that there are item effects here, right? And these item effects, well, for our statistics, we also have to take them into account. This is a little bit, in some sense, toy data because it's very little EEG recording. So yeah, very happy to collaborate on this. And to do proper stats, we might want to have some cluster permutation tests. So advertisement, come to the poster and learn more about cluster permutation tests for LMMs. It's not so easy. Okay, let's have an outlook and a summary. So I talked about these 4 examples here, and these are a little bit like mix and match. So some of them can be combined. You can have deconvolution with nonlinear predictors, etc. Others are more harder to combine, but we're working on it. But this is nice because whatever you need, you can kind of just plug it in like a little bit like a Lego set. There's a long history around this, right? This is not something that I invented in any sense. We just provide one of the toolboxes. More on that in a second. And so this is just a long list of work that I think is important in this regard. And I want to do 2 things. I want to recommend you read these 2 papers because I think they are very nicely showing you the details of it. And I also want to just mention that this technique has been invented multiple times from different people independently, or at least they don't cite other people. No, I really think independently. So this can just happen. So there's also a little bit a need to connect these different literature that people are more aware of them, right? Finally, second last thing, there are many other things I could not, like plugins or tools that I could not talk, like, I don't know, baseline correction, adding functional connectivity into that. Jean-Rémi, maybe he will talk later about back-to-back regression, or maybe not, combining this with decoding, or also work from our group, like you have long and short events or something like this, how we can do that. So I think that's super exciting. There's a list of toolboxes. I will not talk about this because it's on the GitHub. Just check it out, all different flavors and focus, etc. And yeah, with that, I'm at the end of my talk, and I thank you very much for your attention.
A couple of questions, quick one actually. The first one would be that you work with Romy Fromer and O'Connell et al. Response paper. In your opinion, does the central parietal positivity, or CPP, exist as an evidence accumulation signal in MEEG, and why not?
It's really hard. I think it's too early to say. I think what we basically show here is that there can be CPP effects that I personally think are likely because of stimulus overlap. That does not mean that all CPP effects are, that they cannot be evidence integration, but just looking at this ramp, it's not enough to say that this would be evidence integration. But I guess best person to ask is really Romy there. I did a lot of simulations for this. Yeah, that would be my answer. I hope that's fine.
Could the overlap or correction potentially be used to correct artifacts?
Artifact. So it depends really on what artifact is. Potentially, yes. I guess in fMRI they do this quite a lot by adding some nuisance regressors or adding breathing signals, etc. So yes, you can include artifacts into this thing. For instance, in EEG eye tracking, people use regression to regress out eye movements, etc. And probably time-lagged embeddings of this would be a good idea because you don't necessarily know when. You have to be a little bit careful. Like for instance, if you would add a blink signal, right? You could also remove blink-related ERPs or something like this that you might be actually interested in to look at. So you have to make a little bit of distinction whether something is instantaneous, whether it's muscular, for instance, or where it comes from, or whether it's something elicited from the brain, and you're actually interested in it.
Great. Thank you again, Benedikt, for the great presentation and your thoughts. Now we're moving to the next speaker. So after having modeled linear and non-linear effects, let's move to another methodological field that has aroused an increasing interest in the community in the past years, deep learning. So our second speaker, Jean-Rémi King, is a CNRS researcher at École Nationale Supérieure in Paris, and he's currently attached in Meta AI. So now he's going to provide us an overview of the progresses made in the use of deep learning methods for MEG decoding.
So I'm leading a team at Meta AI, where we focus on the interaction between neuroscience on the one hand and AI on the other hand. And I'll try to show you in this quick talk how we can use AIs in two ways as a kind of method on steroids, but also, and perhaps more importantly, as a conceptual framework. So the basic recipe is kind of always the same. We want to use AI system to see whether they learn representations that are similar to the brain in response to the same task. Could it be visual perception or language processing? We want to see whether the AI system that people are developing today and that are typically available on Hugging Face or other platforms, whether the systems learn, for instance, to process language in the same way as a human brain does, at least whether they process these stimuli in the same way. So typically what you want to do is try to see whether the artificial neural networks lead to activation in particular stimuli that are in any way similar to the activations of the brain in response to the same stimuli. Now, because I wanted to be a bit provocative, I thought I should start with fMRI in this event, but I promise that I'll show you some MEG data afterwards. So here is a first study where participants were listening to natural stories in the fMRI scanner, like The Little Prince. As a reminder, maybe just give you a sense of how the experiment goes. This is the type of story that they listen to here in French.
J'étais bien plus isolé qu'un naufragé sur un radeau au milieu de l'océan. Alors vous imaginez ma surprise au lever du jour quand une drôle de petite voix m'a réveillé.
All right, you get a sense of it. Even if you don't speak French, it's a pretty chill experiment, except, of course, that this is in fMRI, and in fMRI you have this horrible background noise, which doesn't make it the device of choice for all questions, of course. But in this particular study that was led by Juliette Millet and Charlotte Koster during their PhD thesis, we compared the fMRI brain responses to these naturalistic stimuli to the activations of an algorithm, which is a bit of a mouthful. It's called Wave2Vec 2.0. And this algorithm is basically a deep net, which is trained to process natural speech, but in a self-supervised manner. So you don't need to have any annotation. You can just give it a lot of speech input, and it will basically learn to predict its own activation. And apparently this is a very good learning objective for it. It's still used as a state-of-the-art neural network for downstream tasks like automatic transcription, for instance. So the whole goal in this case is to try to see whether the activations of Wave2Vec 2.0 are in any way similar to the activation of the human brain in response to natural stories like The Little Prince. And to do this comparison, we follow the exact same recipe as Benedikt introduced. So we use a so-called encoding analysis. In the case of fMRI, the acronym tends to be FIR for finite impulse response model, but it is the same equation as the TRF model in EEG. And basically what you build is a linear readout of all of these activations of Wave2Vec 2.0, such that this linear readout is maximally predictive of the brain response. So here is a cherry-picked example on the top here. You have on the horizontal axis the time from the onset of a story, and the vertical axis is the amplitude of the bold response, of the fMRI response. And the black line is the actual measurement, and the red line is a result of this linear model. So the two lines are not exactly identical, but they tend to go up and down in the same way, which suggests that this linear model captures relatively well the function of this particular voxel in response to particular speech input. And so that means that there is somehow a linear readout of Wave2Vec 2.0, which produce the same type of computation as this particular voxel. All right, so this is for one voxel in the superior temporal gyrus. We can repeat the same type of analysis for every voxels in the brain. And what we observe is this brain map where the color is the correlation between the true amplitude of the that was recorded, and you see that it peaks here in the areas of the brain which are typically associated with audio and speech processing. Because we have a lot of participants here, these are curated datasets that were made publicly available by a variety of labs, but most importantly, perhaps by Uri Hasson and Sam Nastaz. Because we have a lot of participants, the statistics suddenly is not an issue anymore. All right, so this is not interesting in itself. What is interesting to do is repeat this analysis, not using all of the features of Wave2Vec 2.0, but to do this for each layer of the deep net. And so for each layer of the deep net and each voxel, you can try to do this encoding analysis and evaluate the extent to which there is some similarity between the activation of the algorithm and the activation of the brain. Similarity, as I said, evaluated with this linear mapping function. And when we do this, we observe something which is, to me, quite striking. The areas which are associated with early auditory processing, here the primary auditory cortex, the belt, the parabelt, they tend to be color-coded in blue, meaning that their activations in response to natural speech tends to be most similar to the first layer of the deep net. Whereas if you take areas, for instance, in the temporal pole or in the prefrontal cortex, you have a lot of voxels which are color-coded in red, meaning that their activations is most similar or best linearly predicted by the deepest layers in the deep net. And in between, you have this sort of continuous gradient. Right, so that means that basically the comparison between fMRI brain responses and these deep nets leads to a linearly similar hierarchy of representations to natural speech. And this is very striking because Wave2Vec 2.0, like many deep net algorithms, has not been built to work like the brain. The goal is to is kind of an engineering goal for industry mainly. The goal is to have something that works, not something that works like the brain. All right. Now, fMRI, as you know, is cool, but MEG is cooler. So let's do the same type of analysis now with MEG. In this case, what we did with Charlotte Kochte is to do the very same type of method, but in a reading task. And we do the comparison between the MEG brain responses to reading. And we do this comparison between brain activations to 3 different models. A deep convolutional neural networks trained on CAPTCHA recognition. So it's a deep net that presumably only represent visual features. We did the comparison also with word embedding, which is a way to represent word with vectors, typically semantic vectors, but these vectors cannot be contextualized. So the word bank, for instance, can have different meaning depending on whether you mean the bank where you withdraw your money or the bank, the river bank by which you walk your dog. Word embeddings would have the same word vector in both cases because it cannot be contextualized. And the third comparison that we made is between the same brain responses, but now to what used to be called contextual word embeddings, and which is now referred to as large language models. So we analyzed the data that was made available by Jan Matthias Schoffelen at the Dantes Institute, where participants read sentences in a rapid serial visual presentation mode, so the words are being flashed one after the other. It's in Dutch, but I thought it would be good to have English examples instead. And for each participant, we can apply some source reconstruction and infer where in the brain the single trial MEG responses are most likely to occur. We have a lot of individual variability, but on average, what we observe is a typical response. We have, after the word onset, an activation in the back of the brain, occipital areas, ventral dorsal activations, prefrontal ultimately is being recruited, and ultimately the signal seems to die off in the so-called language network, in the temporal lobe and in the prefrontal and parietal areas. Let's do this again. Occipital activation, ventral dorsal stream, premotor activations, global ignition, and ultimately the signal dies off in the language network. All right, so this is just an average response. average response to words. What we want to do is to see whether at the single trial level, the activations that we can infer with this source reconstructed MEG responses are in any way linearly correlated with these three deep neural networks that I mentioned. So the visual embedding, the word embedding, and the language model. All right. So this is the result of the analysis is being plotted here as a function of time. Around 100 milliseconds after word onset, we see that we have the occipital lobe, which is being color-coded in blue, meaning that the activation in the back of the brain tends to be linearly corresponding to visual embedding. So that's reassuring. After that time period, we see that this effect propagates along the ventral and dorsal stream. And now we have a change of color, meaning that in this area, which is known as the visual word family area, we have a change of code. We have both a perceptual or linear correspondence with the visual embedding, but also an additional variance explained by the linear mapping to word embeddings. So that, let's say, confirms what had been shown with factorial designs before. If we wait a bit more, we see that this green effect propagates mainly along the left hemisphere and really peaks around 400 milliseconds, which is known to be one of the hallmark of semantic access in the brain from the earlier work of Marta Kutas and many others since. Sources are not super precise, but it seems to be clearly corresponding to the language networks. Now, if we wait a bit more, we see that there is a lot of back and forth. It's not quite clear exactly what's going on, but if you pay close attention, you'll see every now and then some peaks of red. And that is because now, every now and then, there is additional variance explained by the features of the language model, of the contextual word embeddings. And ultimately, if we wait long enough, the only thing that remains significant is the correspondence or the similarity with contextual word embeddings. So this is great, right? Because by doing this comparison between deep nets and the brain, we recover the classic hierarchy of processing for reading, visual processing stage, word embedding or lexical access, and ultimately a contextual word embeddings in both time and space. All right. So these are examples of encoding analysis where we do this mapping between deep nets to the brain by trying to predict the brain as accurately as possible, given these deep learning features. But of course, we can do the reverse to try to recover the features that AI systems have learned directly from brain activity and potentially use this for decoding. So here's an example that was led by Alex Defossé. The architecture and the data is available online for those who are interested. Here, what we did is learn to align the activations of MEG responses to natural speech that were collected in part with Laura Williams at NYU and team, and the activations of the speech model that I showed you before, Wave2Wave 2.0, in response to the same stories. We use contrastive learning to try to estimate or to find a way to transform the MEG data such that we could recover the most likely spoken sound and the most likely words at each time point. And long story short, it works much better than what I was anticipating at the time. It's still far from being 100% accurate, but it's quite remarkable. So here is an example where, for instance, for subject 3, listening to a story, the story contains the sentence, "Thank you for coming, Ed," and you see that the decoder is accurate for every individual words. For subject 2, it's making mistake. For instance, the word "what" is considered to be more likely than the word "thank," and the word "in" is considered to be more likely than the word "ed." And for subject 1, it's never correct, right? It says that "you" is more likely, "to" is more likely, "author" is more likely. But if you pay close attention, you'll see that the true word tends to be in the most likely 10 words output by the decoder, and there are 800 possible words here. So it's quite remarkably above chance and quite surprising that we can recover these things. Now we're pushing this with Stéphane Dascoli now in the team to really focus on semantics, and we can show that we can really start to decode the semantics of individual words. So for instance, when the participants are hearing the word night, the most likely words which is being decoded is night, but the second most likely would be evening, and then hour, week, which share some semantic properties. Same for the auxiliary verb would, which otherwise would have been predicted as could, add, will, and so forth. And you can find a lot of examples, like, for instance, extraordinary would be decoded as extraordinary, but the second most likely guess by the decoder would be uncompromising, exceptional, and so forth. All right. Now, just to show you that this is not specific to speech, we also applied the very same architecture, in fact, with little to no modification, to data where participants were watching images. This is data that was collected through the Things Initiative, led by Martin Hedbart, where participants are watching many, many images one after the other. And we learned to align these MEG brain responses to the features of computer vision models. And then we fed this prediction to image generation models. So you, instead of conditioning the generation of an image, let's say by a textual prompt, here you condition it by a brain prompt. And the images that are being generated they're far from being perfect, right? It's, in fact, they are, if anything, always weird and inaccurate. But they share a lot of low-level and semantic features in a way which I think was quite surprising. So, for instance, if you have a panda, the generated image is a black and white bear, which doesn't exist. If you have a person wearing a tuxedo, it still creates a person, but not wearing a tuxedo. If you have a lab equipment, it creates this monstrous lab equipment, and so forth. So it's clear that there is a lot of progress that can still be done, but still the signal that seems to be that we can recover is still it's still quite surprising. All right. Unfortunately, or perhaps fortunately, I don't know, depending on where you're coming from, this is not at the level yet of ultra-high field fMRI. So this is now the same pipeline, but using 70 fMRI responses. This is the data coming from the natural scene data sets. And you see that the images that are being reconstructed are far more precisely similar to those that the participant actually watched. Okay, so the bottom line is that I wanted to show you here that we can use this AI really in two different ways. One, which is just let's say the TRF on steroid, right? Just like as a pimped up method to try to denoise or extract signal. But perhaps more importantly, the reason why it works is also because these AI systems tend to learn representations which are similar to those of the brain, and that creates a really exciting avenue for future research. Right, I think I'm over time, so I'll just want to highlight that this is possible because of a team I'm really very grateful to work with. Thank you all for your attention.
Thank you, Jean-Rémi. Sorry for the delay. So sure. Now we're going to see like these questions that are super interesting. The first one by Robert, if I remember correctly, refers to the analysis. So do analysis like this always need large data sets with more than 100 persons or MEG data? And if so, what is the consequence for all those labs that are typically doing studies with a very few number of subjects?
Yeah, that's an excellent question. So no, we don't need to always have 100 participants. What we are doing at the moment is try to establish scaling laws, so to try to see how better the signals get or the results get as a function of the amount of data that we put in the system. One of the things that we're starting to see quite clearly is that it's better to have more data per participant on a small number of participants than having a lot of participants with a small amount of data per participant, right? So if you have the same data budget, like spend it on a few participants that, with many hours than a lot of participants with a few hours each. The consequence, I think, for the fields, my sort of desire is that we will, as a community, really push even further the sharing of data so that we can pre-train or put together different experiments that were collected in different labs. Because if, I don't know, the lab in Berlin works on black and white images and a lab in Paris works on colored images, I'm sure that you can still benefit from putting the two data sets together and try to improve your modeling of the brain response to images in the first place, while keeping the individual differences. So the hope is that year after year, we'll be able to fuse basically these different data sets together and perhaps unify this the whole research. Yeah. But that's that's a desire. I don't know whether we'll get there.
Thanks. Now we have another question. Could passing spatial location of the magnetometer into the brain module of MET decoding pipeline improve performance? In particular with the R by exponential C by T dimension that ignores where on the scalp the codings were from.
All right, that's a technical question. So yeah, I skipped over all of the details for this. I also don't think that it's not just a play of hands. I think it's also important to keep in mind the simple part of the story, because I think these technical details that we have, they are very likely to move very quickly over the iterations. In the pipeline that we have, how do we train these deep nets? When you train a deep net with just one person for 1 hour, it doesn't work. You don't have enough data, you have too many parameters, it doesn't work. And so you need to sort of pull together the different participants. So one of the things that we do is that we inform the deep net about which participant is being to which participant a given trial corresponds. So we have what is called a subject embedding layer. But we also, because we have different devices, we also we also observe that informing the deep net where the channels are with respect to each other improved the ultimate modeling of the data. So this is also something that we add to the deep net. So I think I see this as an extension of the mixed model analysis, right? In the mixed model analysis that Benedikt told us about, you basically try to keep these different elements. So you have the subject variable, you have the item variables, and you want to keep those in your statistical model to improve your precision. And here it's kind of the same. You want to say, okay, I know which subjects are being recorded, I know what device they're being recorded with, and I'm going to try to squeeze this information to the deep net in order for it to be more precise. So I don't know whether this precisely answers the question, but the idea is that these engineered layers are likely to change, but I think the spirit will remain. Yeah.
Great, thank you very much. I think that it's time to move to the general discussion with all the speakers. Thanks again for your presentation, Jean-Rémi.
Thank you.
So yes, after having heard these two great talks about specific methodological tools that are now developed, I think it's time also for us to take a few steps back and to consider broader questions related to your respective field and their potential impact on the society. So while we wait for the first questions posted by the attendees, I wanted to launch the discussion to discuss about specific points, in particular with how we can disseminate knowledge. And given your expertise, in particular in open science, I was wondering which kind of advice would you provide to help researchers and any person interested in your field in disseminating their research? Because we have, for instance, the eEG pipelines initiative. That is a great start to also improve the producibility of the research and also to share tools among researchers.
So for sure, I think one way that we can build this kind of or make this more accessible, right, is to build communities like CuttingEEG. I think is really nice for that, these kind of conferences, and also build toolboxes. So this is something that I do a little bit more. And of course, I mean, Robert is sitting here next to each other. He's one of the arch examples for building toolbox to make these things more accessible, right? So I think that's really important to share this. And yeah, I mean, I guess everybody then can look up what kind of analysis or specific questions they have themselves in the data, as long as these tools are easily usable, available, etc. But then, of course, there's also the flip side that people have to maintain this, right? And yeah, I'm very thankful for a field trip, EG Lab, M&E. I mean, this would be a pain without these toolboxes.
Maybe I can have a go. Maybe like the very classic thing, right, is to promote open source and open science in general, making sure that the data can be shared. But I think everyone sort of know this, especially in this community, in this conference. One thing which I think is quite clear is that we're not at the level of the machine learning communities which systematically share code, or where you can, well, except perhaps for some industrial labs, but in academic lab, at least, it is systematic that you can reproduce the experiments when you submit to a conference. And I think the reason why this is not the case for us is is a bit of it's a bit cultural, but also because it's a lot of work. Submitting a paper without having to submit a clean code is a lot less work. So I think what we need to do is to create incentive, like awards and I don't know, things which would help incentivize the open sourcing and the replication of studies, and that would be my advice.
Thank you very much. Another question we would have with the organizers is about the impact of AI. How does AI impact personal autonomy, privacy, and potentially cognitive freedom in daily scientific practice? And also, it's also linked in an indirect way with you work, for instance, Jean-Rémi, on speech decoding, and also with my field with BCI, in which we have to classify and to translate one activity into commands, and sometimes some ethical questions can be raised.
Maybe I can start because I talked a bit about speech decoding. So there are a few things. So the first thing I'd like to say is that this is an important ethical consideration to keep in mind and to make sure that we continue having discussion on. I am not too worried about where things are going on the BCI part, because I do see that the decoding of anything remains extremely challenging, even in the very best sort of scenario where you have like 40 hours on a single subject, making sure that he's not blinking or having any eye movements. And this is so hard to even decode something then and on the perceptual side, then to me, like, there is nothing that suggests that we will we do have a technology where we could decode inner thoughts that would cause ethical problems. That being said, we need to be vigilant about what's possible. It's also important for that reason to make sure that these research remain publicly accessible and reviewed by the scientific community. But now there is this issue of AI for neuroscience specifically, but there is the issue of AI much more generally on whether the algorithms are opaque or not opaque, whether the data is being stored in a place where perhaps the regulation is not quite identical. Like, this is a much broader discussion, which I don't think would be specific to the analysis of EEG.
Yeah, I had a question for you, Benedikt, about the sustainability and how sustainable practices can be integrated in the topics we discussed today, and about how we can also perform and carry on our research project in a sustainable way.
Yeah, I guess it's hard. It takes time, right? Jean-Rémi already said that publishing a paper with not clean code is easier than publishing it with the clean code, right? So this has to be somehow there has to be incentives, and I think prizes are cool and great, would be nice to have, but also I think more systematic things. So at least I'm in a computer science department right now. At least there, people start to hire research software engineers, for instance. I didn't even know this term existed half a year ago or a year ago. Then I actually gave a summer school on it. And research software engineering is basically all this toolkit of making sure that this your tool is doing what it's doing. You have tests, you have documentation, you share the code, you make it available. These kind of skills, I do think they're super important. I think that there should be a systematic, should somehow be, I don't know how it can be supported, but maybe every lab should have one RSE person or something like this, just to make sure that these things will be sustainable in the long term. But there's also, like, this is the one meaning of sustainable, but then of course there's the other meaning of sustainable that I think Anne Uray is talking more on Wednesday. Is sustainable also in a sense of economical sustainable, because not everybody can train their GPT-2, and let's, like, as soon as cynic person would say burn a forest when doing it, right? Because there's a lot of energy needing for these things, and I think we have to move to more efficient algorithms. So this is some part of the things that I do. I try to improve the tools I use, languages and possibilities to just make it more efficient, not do unnecessary computations, etc. I'm not sure if prevention is really scales very well or will be the thing, but nevertheless, it's a small thing that I can do. Yeah, it's a trade-off, right? It's always a balance, I guess.
It's true, because for instance, in particularly for computer science, I would also like this race in like increase just about a very tiny percentage your accuracy score, and you're happy of that, but it requires, like, I don't know, like days of GPUs to be able to reach this this score. So there is also this concern about it. So is it actually does it worth it actually to run such a huge computation for just very few improvements? And also linked to that, just I wanted to mention a tool like MoeBB, model of all benchmarks in BCI. Sorry, I'm super biased. Okay, in BCI, sorry. But in this tool, what I really appreciate is that they also provide some assessment of the CO2 emission linked to the classification algorithm. So I think that there is also a way to assess our footprint. I think it's also a way, perhaps the starting way to see whether we can change our practice. Okay, I think I can go to the next question about epistemology for Jean-Rémi. Yeah, about different ways of thinking, and if, according to you, what would be like the different, sorry, people are just writing at the same time, so complicated. So how can sustainable practices can be integrated in the topics we have discussed today, and perhaps also, sorry, some people are writing now, I'm very sorry. So, typically, if there is a different way of thinking about your own research project, I mean, having like a different perspective in dealing with your work, and if there is epistemologic frameworks emerging from this research.
Okay, so maybe one point about the sustainability. I take this issue at heart. I'm also very passive in a way that the AI race is happening with or without us, in a sense, completely. Especially in neuroscience, we're not I think in the core business of AI. One thing that I think we should try to promote is making sure at least that the model that results from these very intensive training at least remain available to the public, so that it's not burned for nothing, in a sense. Epistemologically, this is a huge. I think to me this is one of the revolution of AI. It's not just how like having this crazy tool that can do a lot of things. Is even how you ask a question in the first place. Very concretely, for instance, when we do. When we compare what we used to do like 15 years ago and what we do now because of AI, we used to compare models not only on goodness of fits, but also on the extent to which they had many parameters or not. Right? We use like this AIC and BIC type of model comparison. And to me, this has gone now because of AI. Like we have like billions of parameters, so we cannot just do the comparison on the complexity of the model, and so the sole evaluation suddenly became on cross-validated prediction, and that's sort of switch a bit the focus on how you address a question. That's one issue on epistemology. And the other kind of revolution is that just as an anecdote, perhaps, when I was writing grants on neuroscience and AI 10 years ago, like I should try to summarize like the best quotes, but I had so many criticisms. Like people were saying, this is like these deep nets, they are black boxes. We don't know what they're doing. They don't even manage to recognize an elephant from a giraffe or whatever. Like, what do you want to compare this to the brain? And this has completely switched now, because we do see that not only they're really good at tasks, but they're really good at them in a way which is useful to study how the brain functions. And so that's, I think, arguably something which is surprising. At least many people in the field were predicting quite the reverse. And that changes the way we build model of the brain, whether we do them just from sitting in an armchair and reading papers, or whether we, like, let's say, fit more and more data, as well as think about these papers. So the way we approach the question of how to build a model of the brain has changed, and I think will continue to change in the future. So that's quite interesting to witness.
Thank you very much. Yeah, now we also have like also the questions about scientific integrity, deontology, especially like what are the key ethical considerations or challenges in maintaining scientific integrity and transparency in this research. And of course, yeah, when you're talking about meta AI, also there is a rising question about the private companies. What are like your own considerations in this case?
So I would ask, what are the considerations that we should not be considered anymore?
Exactly.
It's as before, right? So it's being able to make sure that the research is not let's say guided by an underlying agenda, making sure that it's replicable, accessible to the public, that it contributes to create common good. There is I think there is an issue that deserves to be discussed on how the private sector contributes to the building of knowledge. The bottom line that I personally chose, but I do appreciate if it's not shared by everybody, is that as long as the private sector allows us to publish in peer-reviewed journal and release the results of the research, at least we stepping in the right direction. But I think that this is not a closed argument. I think this is something we have to remain vigilant about, and making sure that we're not being utilized in any way.
Benedikt, any thoughts?
So I don't know. This is something that a week ago we already briefly discussed, but I just noticed in computer science. So since switching from the Donders to a very cognitive kind of idea to computer science, it slowly creeps in that I do more and more computer sciencey things, right? So it's like it's never purposefully. So it's a little bit hard to stay on track if everything else is revolving around something else. And I guess in meta, this will be kind of similar. And what would worry me, like why I really like to be in a public institution, is that I mean, tomorrow basically somebody could say, yeah, you're not gonna publish that if you want to keep your job, right? There's always somebody on top that could stop the publication. And I personally shy away from that, but I totally understand. I mean, the resources so are different, and there's many benefits as well. And it's yeah.
No, I agree. I think this is there is a risk here. I think what we have to be even more conscious of the impact even within the public sector, right? Because as professors, or when you're a PI, you're also training young students, and they're not all going to stay in academia, right? So we are also training a cohort of people who will eventually work in industry. So I think it's unfortunately more gray than what we would wish. My personal opinion is to try to, at the very humble level where you work, try to push in the direction that you think is best. But I certainly agree that these are not easy topics.
Thanks. Perhaps, yeah, I will have like a very final question, but quite huge one. So that's why we need to spend some time. Yeah, it was like about perhaps next generations of researchers in particular. So we had like very interesting question about the skills that should be taught to the neuroscientific students, and to adopt more complex analysis methods, and also whether the use of like this tool like ChatGPT would be advisable or not. And linked to that also, perhaps just a small advertisement about the session of tomorrow about the careers in neuroscience, chat by this wonderful and unique Anna Assurance here. So yes, I think it's great also to think about different skills to be taught to the next generation. So I would like to know your thoughts about it.
Benedikt, do you want to start?
Sure. So I have multiple thoughts on this. I mean, of course, it doesn't scale. We can't teach everything. It's impossible to know everything. I think Robert really phrased it nicely. So these complex analyses require new skills. I do, like, at least in my toolboxes, I try to make to abstract away all the Like you will never see design matrices or something like this. All the technical, you should know about them, but you don't need to know exactly how they're built, etc. The hard part, this is always what I try to keep in mind. The hard part should be an interpretation of the data. So you need to know what you can do, what you are doing right now, and what does it mean, right? And then all the technical stuff, I think this can be to some degree abstracted away, but it's going to be very hard not to teach them the basics. And then something has to give. And I was just in my mind thinking, okay, what could we not teach? And it's not easy, right? It's not easy. Maybe it pains me also a little bit, but maybe like the details of filtering is maybe I think there's nowadays really agreed upon what kind of filters we should use or so, and it's really hard to, like, you need to know a lot about filtering to come up with your own thing, I think. But okay, nobody's staring at me anxiously, so that's good, I guess. But I don't know. So maybe there are some topics where we could say, okay, this is now more standard. We will skip over this a little bit faster. But then we spend more time on statistics or on, I don't know. Also deep learning, I guess. In computer science, people are really starting to export deep learning for beginner courses, etc. But it's really hard to do, without being just clicky clacky, which is also fine for some, I guess.
This would be thoughts about the interpretability of the methods they're using, because there are super sophisticated methods, such as in deep learning, for instance, but not only. I'm not just blaming deep learning, but sometimes it's hard to interpret them. And okay, you might have like your tool already ready for you, but then understanding why it's working or it's not working, which kind of data you're manipulating or not, it can be tricky and requires time and expertise or so.
Yeah, no, I agree. I think interpretability is a very interesting point. And Jean-Rémi earlier, he said, okay, people don't count parameters anymore. And this kind of, there's like this, there's a famous paper that I forgot the author of, but there's two ways of statistic. One going more for inference, counting parameters. Still, this is more where I would see myself. And then there's more the prediction kind of thing, where you just throw in everything you've got and you regularize it. They're not necessarily exclusive or so, but I think they're worth both different. If you have a little less parameters, like whatever parameters, so degrees of freedom may be better, it is easier to understand, and it is less black boxy. I do think the black box argument still is there. There are some methods, but it's very difficult, right? But already SVM or so is very difficult to understand. So you don't need deep learning for that, I guess.
Yeah, I agree, and I want to double down on this. These are not mutually exclusive, but now there is clearly a new trend that at least I was not trained with before focusing on prediction. I don't have a general answer to this question of how we should teach to them, but I can tell you what we do in the team. It's always a kind of gradient of complexity. So first, like on every project, it's looking at the average response and then doing some linear modeling analysis, like linear decoding or linear encoding, and then only fit a deep net on top. And I think this is critical, because if you go directly to the deep nets, then it's 100% sure you'll make like stupid mistakes on how the data has been split, for instance, or like the impact of baseline corrections or whatever. So I think it's really important to not jump directly to the deep learning most complex methods first, and also perhaps for the youngest audience, I don't think it matters to be at the cutting edge of AI, because this is moving very quickly. So it's fine to just use what used to be developed a couple of years ago, because we're not even in the regime where we have enough data, or at least not enough data at the right quality to use the latest deep net. So I think it's okay to be a bit behind the AI race, in a sense.
Agree, and perhaps linked to that is perhaps the most complex tool and stuff would be to pick the right tool adapted to your scientific question, not because like it's fancy or trending, but actually it will actually answer your scientific question because of specific some limitations, but number of samples or that kind of data you're manipulating. Perhaps one method is not super sophisticated, but would be more suited than another. And perhaps the trick in this kind of knowledge to
I'm not sure I fully agree with this. So this is a very classic effect, right? Know your question first and design your experiment accordingly. But at least in my experience, it's quite the amount of things I've learned during the process of analyzing the data and reshape the question accordingly, not just in a cherry picking fashion, but also sort of discovering something I wasn't thinking of originally is quite important. I think it's rare are the projects where I had a very clear question and this is the question I answered ultimately. It's more like a back and forth discussion between the data and the theory in a sense. So I don't know. Yeah, it's true that if you know the question, that you'll know which tool to use, but it's kind of a discussion between the two, and so I think you kind of need to learn
I agree, but I think it depends on the type of data you're manipulating also. That could also guide actually. I don't see like a limitation, but actually a way to reduce like the amount of tools you could use just to help you and to guide you a bit.
Yeah, I agree.
Awesome. I think that we are almost in the very last part of this session. It's the time also to close the session and to warmly thank you, Benedikt and Jean-Rémi, for presenting your work, of course, but also having shared your thoughts. And I think it's really important, especially for the youngest generation, to have also your thought about this kind of questions that could be for the next year more important. I would also to thank the attendees, and I hope that you enjoyed this session. All the materials and some additional surprises will be available in the GitHub page as we put in the on the forum. And finally, I would like to thank the CuttingEEG Dream Team that helped me in the organization of this little bit hectic session. And a special thanks to my dear friend Anaïs here again, as you can see, who will share tomorrow this session about careers in the after the PhD. And I think you should come because it will be super interesting to see different panel of profiles. Have a great day all, and thank you. Bye-bye.
Thank you all. Thank you, team.